Hello, we're glad you've joined us for this live webinar, Taking Control of the Unknown, Nanoparticle Hazard Containment. I'm Judy O'Rourke of LabRoots, and I'll be moderating this session. Today's educational web seminar is presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration and learning. It's brought to you by LabConco. Serving the scientific community since 1925, LabConco manufactures laboratory equipment, specializing in ventilation products such as chemical fume hoods and biological safety cabinets. Other product lines include glassware washers and freeze dryers. LabConco has ISO 9001 certification, one measure of the company's commitment to quality and consistency in design and manufacturing. LabConco equipment is manufactured in Kansas City, Missouri, and Fort Scott, Kansas, and marketed worldwide to academic, industrial, life science, pharmaceutical, environmental, forensic, and clinical laboratories. To learn more about LabConco, please visit www.labconco.com. Let's get started. This webinar has been approved for continuing educational credits. Please click on the CE button at the bottom left corner and follow the process to receive your credits. You can post questions to the speakers during the presentation while they're fresh in your mind. To do so, simply type them into the Q&A box, which will open when you click the green Q&A button at the lower left of the presentation window. Questions will be answered after the presentation. To enlarge the slide window, click on the screen icon in the lower right-hand corner of the slide window. If you experience technical problems seeing or hearing the presentation, just click on the support button found at the top right of the presentation window, or report your problem by clicking on the green Q&A button at lower left. I now present today's speakers, David Wasesha, a product manager at LabConco, and Matt Hull, PhD, a senior research scientist at NanoSafe. David manages LabConco's particulate containment enclosures for nanotechnology and pharmaceutical applications. He earned a microbiology degree from the University of Kansas. Although a microbiologist at heart, David has also worked in pharmaceutical analytical chemistry. Other products in David's portfolio include water purification products, glassware washers, and glove boxes. Matt has more than a decade of experience exploring the applications and implications of nano-enabled technologies. Prior to founding NanoSafe, Matt held an appointment on the Nanotechnology Technical Advisory Group to PCAST and served as Senior Research Scientist at Luna Innovations. In 2003, Matt developed the concept for the NanoSafe framework, which provided an integrated risk management strategy for nanotechnology facilities. This framework eventually led to spin-off programs such as nanotechnology waste recovery and recycling processes, life cycle eco-toxicological studies of nanomanufacturing, and nanomechanical sensors for chemical and biological agent detection. Matt earned his PhD in Civil and Environmental Engineering Biology from Virginia Tech. Matt Hull will now begin his presentation.
Hi, everyone. Thank you so much for joining us. I want to tell you a little bit about sort of the first part of our webinar today. I also wanted to clarify I have a dual affiliation, so I'm president and founder of NanoSafe, which is a small nanotechnology safety provider, and I'm also Associate Director for Innovation and Entrepreneurship at the National Center for Earth and Environmental Nanotechnology at Virginia Tech. So I wanted to go ahead and dive in here. I want to tell you a little bit about, some of you folks may or may not know some of the background here about nanotechnology and why there are concerns about containing engineered nanoparticles to begin with. So I want to give you a little bit of an overview of why we're concerned about these materials at all. First off, what is nanotechnology? The U.S. National Nanotechnology Initiative, or the NNI, set some guidelines here for us for what we can consider nanotechnology. So typically the size range 1 to 100 nanometers, and novel properties or functions that stem from that small size. And as as science and engineering have progressed, we can now manipulate and control matter on that atomic scale. So this is what we define nanotechnology as. Why is it so different? So at the nanoscale, size and surface area start to do some funny things with us. As particle size decreases, the fraction of atoms and other molecules at the nanoscale that are near the surface increases. And these surfaces can drive reactions. They can also have potential toxicological implications that have to be considered. So as these particle sizes shrink, these surfaces and their potential reactivity becomes a greater concern for us. It's not just an issue of size and surface area, but the fact that properties change. So as we shrink gold, for example, it goes from the nice gold color that we've come to know and expect from gold to, for example, a ruby red color that is attributable to the interaction of these nanoscale particles with light. So not only is size, surface area, those types of things becoming an issue, but now you have unique behaviors that we don't tend to know about or expect from these materials that we've known at the bulk scale. There's also a difference in the engineered nanomaterials versus natural or incidental materials. The materials that we're engineering in the laboratory, that we're controlling their size and shape, we're working with different elements from the periodic table. We're creating unique functions, sizes and surfaces, those types of things. Those are very different from the types of incidental particles that we encounter in nature. In particular, the concentrations that we would experience of engineered nanomaterials could be a major concern, particularly in manufacturing environments. And so there you see at the bottom of the slide just differences in some naturally occurring nanoparticles such as viruses. We also have diesel exhaust and some of the particulate matter that we tend to be concerned about from combustion or welding activities, those sorts of things. Those are what we would consider anthropogenic in nature or human created, but also they're incidental. And we're specifically focused here on the engineered nanomaterials, for example, buckyballs or metallic nanoparticles, those sorts of things. Just quickly to go over some general classes of engineered nanomaterials and their applications. The classic nanomaterial buckyballs, carbon fullerenes that are often used in pharmaceuticals, photovoltaic devices, for improving the harvesting of solar energy, carbon nanotubes that are often used in composite and electronic devices. My guess is that a lot of folks who are interested in nanoparticle containment may be working with these types of materials. Metal oxide nanoparticles, which are often used in cosmetics, catalysts, antimicrobial agents. Nano silver, for example, is a really big, well-known metal oxide material. Those are a really broad class. Semiconducting quantum dots, those are sometimes used in displays. Television sets these days have those, or may have those in certain brands. They're also used in biological probes and also pharmaceutical agents. If you want to learn more about where to find more information about these different types of nanomaterials, there are a couple of nano inventories out there that include, this is the Consumer Products Inventory. There's the website for that you can see. This was developed by the Woodrow Wilson International Center for Scholars, and it is available to search and see what's out there. There's also another database in, I believe it was Denmark, that created this, and it is also a very good resource for understanding a little bit more about nano-enabled products. So why worry about nanoparticles? They're uniquely engineered materials, as I've touched on here before. We're not interested in these materials for any other reason than they have unique properties and behaviors or capabilities that we don't have at the bulk scale. And that in and of itself makes them, it gives us a sense of there's something different about these materials that we're paying attention to and taking an interest in. Because of these unique behaviors, or because of these different properties and these uniquely engineered characteristics that they may have, they may behave differently. So size, shape, these things can lead to unique physical and chemical properties that we have not encountered in the past. We're also still learning about them. We don't know everything there is to know about them, and they're still evolving. And what I mean by that are people are continuing to develop different types of nanostructures. They're increasing in complexity. You have folks developing or integrating nano with bio. So this is not something that is going away anytime soon. These are materials that are under constant development. I know at least in the U.S. there's a great deal of funding activity in support of new types of nanoscale materials, but also more complex integrated nanoscale systems. So a lot of you, particularly folks who are working in university research labs, you're going to be encountering nanoscale materials that may be more complex than what we're looking at even today. So nanoparticle transport in gases differs from molecular transport too, and so that's something important for us to consider. Nanoparticles are going to behave somewhat differently in air. They're 10 to 100 times larger than gases. They have greater size variability. More surface area leads to increased surface charging effects. They have more mass and increased mean free path length. And they also have surface interactions that gases don't tend to have. So there are differences here. They're not the same as gases. And NIOSH, who has been the National Institutes for Occupational Safety and Health, has been actively engaged in looking at nanotechnology research as it's progressed from fundamental nanoscale science and engineering into advanced materials, has been very active in this area. And they have rightly cautioned folks to take a closer look at things like respirators, personal protective equipment, and engineered containment systems such as the ones that Dave will be touching on here briefly. So this is something that we need to continually pay attention to. And as this question has emerged, industry came to me and my small business and asked the question, will our enclosures contain nanoaerosols? If you're working with them in the laboratory and you're using these pieces of equipment, will they contain a nanoaerosol if generated within it? And that was a really complicated question to answer, as some of you may have encountered, particularly those working in university research labs. There are a number of questions that followed from this, and you would think that existing testing methods may address this. It's complicated to generate and measure a nanoaerosol. There's interference from ambient nanoparticles. There are nanoparticles all around us. You're breathing them right now. What are the caveats and limitations? What can we say? What can't we say? Does the test add value for the manufacturer to the person using the piece of equipment that's been tested and to others? Is it an affordable test? Folks who've done a lot of work with nanotechnology know that nanoscale characterization is not cheap. It is an expensive undertaking in many cases. And so affordability of a test to verify containment is very important. And again, how do we communicate results to end users? What can we say? What can't we say? And how will folks use this information? These are all questions that have to be considered here. So what we did when we developed this test was we looked at an existing method, and the ASHRAE method that currently exists for looking at the containment of an enclosure of a tracer gas by a containment device is a sort of a proven and well-known standard practice. And it may be imperfect, but it is a way to test the containment of a device and compare its containment to other devices or to some particular level of containment. So we started with this basic test format, which you can see there starting from right. I'll actually start from the right and come over to the left. You have your enclosure. Within your enclosure, you have the tracer gas, which in this case is called sulfur hexafluoride, and you have a tracer gas ejector, and that is just something to aerosolize the gas within the enclosure in some uniform fashion. You have a tracer gas detector and, in some cases, a mannequin that are mounted on the external part of this that would represent the user of that enclosure, perhaps someone weighing out carbon nanotubes or silver nanoparticle powders within the enclosure. And so this is your basic format. What we did is we changed a couple of things. Instead of the sulfur hexafluoride tracer gas, we used a nanoaerosol, in this case silicon dioxide nanoparticles, and a nanoaerosol generator, which was a modification of the sulfur hexafluoride tracer gas generator. We also substituted out the tracer gas detection instrumentation for a nanoaerosol characterization suite. So that's the major modifications to the testing procedure. And there are some major differences there. Sulfur hexafluoride tracer gas, it's obviously a gas, a molecule, and we're dealing with silicon dioxide nanoparticles. Just the process of making the silicon dioxide nanoaerosols is pretty challenging and complex. This is not simply a matter of taking some of your nano powders that you purchase commercially off the shelf and tossing them up in an enclosure and trying to get, or agitating them. You don't tend to get, from our experience, you don't tend to get a lot of individual nanoparticles from that, at least not in a reproducible manner. You may get them in spikes, you may see them from time to time, but you cannot reliably test a device, again, based on our experience, with bulk powder aerosolization methods in these units. We had to develop, we had to implement this method that was published by Michelle Ostrott and her group back in 2008. And we integrated that with the test to replace the sulfur hexafluoride tracer gas. In addition, you can't just use a tracer gas detector, so you have a major challenge in terms of detection of how do you visualize these nanoscale particles. Some of you may be familiar with Abbe's diffraction limit, which basically says that it's not easy to view a material optically below about 100 nanometers or 200 nanometers, and this poses some real challenges. So this is where you tend to run into quite a few or quite a bit of expense when you're trying to do your nano characterization or containment of your enclosure. So it's expensive, it's time consuming, and it's a very challenging place to work. So we have to overcome that. And there's a lot of different characterization approaches available. When you take out those that don't work well below 100 nanometers, you really run into some limitations there. So you don't have a lot of options available to you. TSI in Minnesota produces some fantastic instrumentation that can be used. There are a number of other companies that produce these things as well. And what we ended up going with to help measure and characterize our nanoaerosol was a scanning mobility particle sizer provided by, manufactured by TSI. And this gave us real-time nanoparticle size measurement within a 10 to 420 nanometer window. We can combine this with other techniques to get a broader size range of what's present there. And we can operate the instrument in different modes, scanning, single, where we can look at individual sizes of individual particular sizes of particles, and we can look at concentrations up to 1 million particles per cubic centimeter. This is quickly just an overview of the operating principle of the nanoscan. This is something you can maybe take a look at later if the slides are archived, but this is just a complex process of eliminating the charge on the particles and then allowing us to count them optically. You're basically using a condensation methodology to grow the particle to a point where it can be visualized optically. A major challenge with these types of, or with our test, was just ambient background particle concentrations. You can't do this test just in an ambient environment. It needs to be done in a clean room environment. So we would take an enclosure, bring it into a clean room, and that's where we would perform our testing with the aerosolization of the silicon dioxide nanoparticles, and then also the measurement of the what was present both within the enclosure and outside. And of course, we would get a baseline characterization level of the particulate levels present within that clean room. We followed a traverse path where we would, this is basically called out in the ASHRAE method, where you scan in real time around the opening of the enclosure and at various points that are considered along the seams of the device. And you can also look up at the ULPA or HEPA exhaust area. Since this is a self-venting enclosure, you need to be able to take a look at that as well. So this is where the numbers that you see there around the enclosure are actually where the inlet for our sampler was positioned as we progressed across the face and around the seams and outlets of that enclosure. Looking at some of the data here, this is just a nice overlay of the initial and final particle size distribution for one of our challenges. The mean diameter here this is showing the mean diameter of the silicon dioxide nanoparticle centered around 23 nanometers and at a concentration of about 4 times 10 to the 7th. And this is good. This is a good result because you can't really tell the difference in the initial and final concentration. And that's, if you go back to what I was saying earlier about using a powder or something like that, you could never generate this type of a nice peak for testing the containment of your device using that type of approach. You would need to use something like this. So these data confirm the suitability of the aerosolization method. We take a look here at the in-hood aerosol concentrations, and we see a vertical gradient, and this is due to the way the hood operates. It's taking air in at the face and exhausting it through the top. So the particles that we're generating near the bottom of the box are actually being taken up into the top of the box and then run through the HEPA unit, or ULPA unit in this case, and exhausted out the top. So hopefully that will be air that contains no particles within it as it exits the top of the unit. So that's all these data are showing, is just the vertical distribution of particles within that enclosure chamber. These are the results of the particle traverses. So the movement of the peaks that you see there are a result of moving across that traverse path and hitting those different numbers at the different points along the face and seams of the enclosure. And the important thing to take away from this, though, is that we're below 1 particle per cubic centimeter in all cases there. So this is comparing the background and the sash opening. There's really no discernible difference between what's happening in ambient environment versus what's happening at that sash when we're aerosolizing silicon dioxide nanoaerosols. So just quickly, results, we confirmed the generation of a silicon dioxide aerosol of 23 nanometers in diameter. We were able to find a concentration gradient with inside the enclosure of 10,000 to about 100,000 particles per cubic centimeter. At the sash opening, the sash and the openings of the enclosure, we did not see particles in excess of 1 particle per cubic centimeter. And as a conservative expression of containment, we could say that that was about 99.99% containment of the aerosol challenge. Just quickly, as we have begun testing these units, we placed them on our website. NanoSafe operates as an independent third-party testing firm. So while we co-present with manufacturers, we maintain an independent role in vetting the containment of their units. And when a company like LabConco agrees to do this type of testing and present it to potential consumers, we present our testing results, a summary of them, on our website, which we could share. We can make available to the group if there's interest in that. But you can download a summary report and view a summary listing of the results that were obtained for the units that are actually listed on our webpage. And IEST is a standards or a recommended practice organization that we've been working with to try to take this procedure and move it out to the public where it can be more widely used for testing nanoaerosol containment devices. Quickly, this is just a summary of what the results look like. I mentioned earlier, how do you communicate these types of testing results? It's a challenge. So we've created this format. There's the internal report that the manufacturers get themselves, and then an external summary report, as I mentioned. Just summarizes what we did, what the results are, and it's freely available. There's no cost to anyone. You can go in there and download those reports freely. We do have some limitations. We can never say at this point that an exposure level is safe. At some point, as more data are collected on the toxicological effects of nanoparticles, one could derive some form of an exposure level or threshold limit value to which you could verify containment and make conclusions about safety, but we do not do that. It's also impossible to test all variations of nanoparticles. You have tubular forms of nanomaterial. You have many, many different structures. It's not just spheres, and you have many different elemental compositions. So it's not possible to test all of those. So we have to take sort of a practical approach to what are some tests that we can do, and what type of information can we gather from that, what sorts of conclusions can we draw about containment efficacy. It's difficult to establish exposure limits, as I mentioned, until more tox data are available. This is a voluntary program. It's not required. Companies like LabConco are not required to do this testing. They do so on their own and as the needs from clients suggest that there's a need for that sort of testing. So, and this is no replacement for regulatory guidance. So with that, I will wrap up my portion of the talk and hand it over to Dave.
Thank you, Matt, for your presentation. We will now hear from David Wasesha.
Well, hi, Matt. Thanks again for sharing your time with everybody. And as Matt has kind of outlined, what he's doing right now is clearly a unique component of the EH&S and industrial hygiene assessment of Nano in the workplace, and he helps us understand, that we're providing enclosures that are going to be able to suit the containment needs of different applications, and certainly as Nano continues to bud, we're experiencing a lot of discovery of new components that we don't necessarily know how they impact the health of operators involved in their handling. That being said, I'm going to cover briefly just a little bit of background on two enclosures that we make for nanoparticulate handling. The first one is our Expert Nano enclosure, which is a modified balanced enclosure that's intended to allow for very easy workflow and hands-on involvement in nanoparticulate procedures. And then I'll also just touch briefly on the glove box on the right, which is our protector glove box. Both have been tested against the method that Matt's group has pulled together, and we'll talk about some of the things that are important about these devices, and certainly any device that you would be using to contain nanohazards when handling them in an open state. So the first thing to really get into is the priority of control, and that really breaks down into two components. The first one is to have a goal to isolate a source, so isolate something that's being generated, and then the second one is to capture that nanoparticulate. So on the isolate source side, we'll want to consider containment, the enclosure and a physical barrier, sources that will allow us to protect ourselves and really create a barrier between ourselves and the hazard. And once we have that established, then we can worry about capturing that particulate and putting it into an environment where it's no longer of hazard to people in the environment where work may be taking place. So that's using a filter, ensuring that the filter is properly disposed of, and when doing so, you're not exposing yourself to all of the things that it's collected over its life cycle. So in short, the easiest way to go about all of this is to use a little bit of airflow. So here's a cross-section of our balance enclosure, and this is effectively showing what is done when air enters this cabinet. So it's a negative pressure device. Negative pressure is a good thing because we can keep particulates pulling away from us, away from the operator, and towards a filter that will capture those hazards. Also, an important part of containment, especially with things like nanoparticulates, is ensuring laminar flow as much as possible. So as you have turbulence, you have resultant eddies that can bring particulates back out of the cabinet, back into the work area where someone might be standing or sitting, and ultimately back into their contact with their body, whether it's on skin or through the respiratory tract. So if you combine negative pressure and laminar flow, containment is really easy. You just need to trap that particulate on a filter of some kind. And so there are a couple different methods, but most commonly we use either a HEPA or an ULPA filter, which I will discuss later. So I mentioned negative pressure. Well, what is negative pressure? So to illustrate this concept, here is a basic household fan. And you can see behind the fan there is negative pressure, indicating that the air is being drawn towards that fan blade. As it's drawn towards that fan blade, it's being sucked towards the source of movement in that fan. So if you think about placing a fan near a window to exhaust a room of hot air during a hot summer day, all of that air will eventually go towards that fan blade or that source of motion. Once it's on the other side of the fan, it's under positive pressure, and it's a little bit more difficult to control with just a fan blade present. So we prefer to keep hazards or things that are dangerous to operators or users under negative pressure as much as possible. As air moves forward to that fan, you can see it's being pulled and then being pushed. There's a lot more control that can be had over pulled air than pushed air. So hazard control is best done under negative pressure. And this is the same concept that you'd experience with a traditional laboratory fume hood, where you have air pulling away from you into the hood and to the outside, pulling hazards away. No different concept. Where things change a little bit is ensuring that the air entering that cabinet is as smooth or as laminar as possible. And what you get when you provide a cabinet with laminar flow is you get an airflow that's moving without turbulence. So as I mentioned, eddies are going to cause, result in swirls back towards operators' faces. So reducing that airflow into a laminar fashion is the most important thing once that air has entered the cabinet. So if we put all these things together and consider what we should have in an enclosure, the first thing is something that's going to provide user protection, or something that's under negative pressure, pulling air away from the operator. Also, we want to make sure that it contains hazardous powders, particulates, nanomaterials, nanotubes, whatever they are. So we want to make sure that it's got some kind of filtration device. Another consideration is to make sure that the device, the enclosure, is going to be made out of a material that's suitable for handling sensitive static powders. Nanoparticulates and other powders, things that have been lyophilized, they often become very staticky, and that can be very difficult to control. So by providing an enclosure that's made out of steel, made out of glass, you can reduce the static as much as possible, prevents any kind of uncommon behavior where a powder might stick to a surface rather than go to the back of the enclosure and then be caught on filtration. There's also some things you can add specifically for nano that help containment. So an ionizer to further assist with static in the air column before something would even get to a wall or the back of the hood. Also airfoils. Airfoils are a way that we can ensure air entering the cabinet remains laminar and without eddies. So if you take a look at your fume hoods in your lab, or a biosafety cabinet, or any kind of ventilated enclosure, it should have airfoils that help direct air into a laminar fashion into the cabinet. There are a lot of other things you can buy that are not necessarily integrated. So, like for instance, a lot of balance manufacturers will make anti-static devices that can assist in this removal of staticky behavior of particulates. So another great one, of course, is, like Matt outlined, having a unit that's been known to contain or tested to contain against nanoparticulates. Really the best way to go about doing that is using the NanoSafe approach. You can certainly use representative tests using powders and gases, but if you're handling nanoparticulates, you may as well just be using a test design specifically for that situation that you might be experiencing when you have employees working in these devices. So once you have a hood, there are a couple things you can do with it. Oftentimes it's just recirculated into the lab. That's okay if you don't have any hazardous chemicals. So if you're just working with nanoparticulates that don't have an odor or a vapor that they're emitting, then you can simply connect a tube to the top of that, and it will allow you to I'm sorry, if you were recirculating and you're not working with hazards, then you could certainly vent the enclosure back to the room. If you happen to be using chemical vapors, our nanoparticulate hoods, whether it's a glove box or a small balance enclosure, can also be vented as well. So even though you think, well, I've got some chemicals at play in my enclosure, I should just use a fume hood. Fume hoods don't necessarily have the best design to handle nanoparticulates. Filter, potentially a higher inflow phase velocity that causes some turbulence. So these nano-specific enclosures can certainly be modified to be used with those chemical vapors. Another thing to really consider is, where are you putting it in this lab? A lot of laboratories or building spaces are really cramped. Sometimes clean rooms are overstuffed with equipment as well, so it can be challenging to figure out a good suitable place for an enclosure. But you have to remember that this is a device that's manipulating air, so the last thing you want to do is put it into an environment where it's going to receive external turbulence. So away from supply air diffusers in the ceiling that are not laminar, away from doors, heavy foot traffic, and of course away from windows if you were to have an open window in a lab, but obviously that's a little bit less common. Vibration is another really big concern. So a lot of these enclosures are receiving ultra-sensitive balances to measure ultra-fine particulates like nanotubes. And so you have to take into consideration the overall stability of the device. Now, we build devices with vibration reduction in them, but that doesn't necessarily mean that they will overcome, heavy vibration from maybe a nearby wall that's receiving a lot of vibration from shifting winds. Also, another consideration is as you move up higher in a building, there's more sway structurally, so it can add to some of the vibration issues. Matt gave a little bit of talk about IEST. IEST is also a great source for nano planning. So sometimes, if you're working in a nano facility, there was maybe 6 months to a year of studies for vibration. So those things can be really impactful and can sometimes have deleterious effects on enclosures despite the engineered designs and construction. So we make a little bit of a discussion about how you should properly do this install for each one of your hoods, whether it's a glove box or a small balance enclosure. But again, the biggest thing to remember is that they're containment devices that are manipulating airflow. So in most cases, you want to make sure that you're putting them into a well allocated area, not by a door, not crammed into a corner where potentially a lot of people are moving by. Also, a couple of things to point out. So when you're working with these enclosures, you want to work as far back away from the opening as you can. In a glove box, that's a little bit different because it's a sealed atmosphere. But obviously, in any kind of hood, whether it's a fume hood, biosafety cabinet, powder enclosure, or nano enclosure, you want to work at least 4 to 6 inches back into that hood. You also want to make sure that you check these hoods every year at least. So that involves in a nano hood, scanning the particulate filter, the HEPA or ULPA filter, and of course also making sure that you have test data available from someone who's performed containment analysis on that hood. You want to know that it's doing the job that you've purchased it for and put it into place for, not just to sit there and look nice in the laboratory. You also want to think about PPE, so moving materials in and out of the hood, handling the materials inside the hood, gloves, whatever might be needed, consider those as well. Another big one to keep in mind is, unlike a fume hood that may potentially be operated with a sash wide open, in a smaller enclosure, especially a powder enclosure, keeping the sash down ensures that operators don't increase the area where there's a risk of being exposed to the particulates that they're handling. We've asked NanoSafe to test our hoods in a certain way with those sashes down, so certainly any data that they produce is contingent on proper use of the hood, and so that's an important thing for users to keep in mind. Another one, you read the user's manual, read the brochure, learn a little bit about the device before you go to work with it on a daily basis as a protection tool. And also, if you're using any kind of nanoparticulate, before you're shutting the device off or turning off the airflow or maybe opening the door to a glove box, make sure that you properly cleaned up the inside. All right, so that's how we isolate a source, but how do we capture it? This is probably the more involved part of the enclosures, because these are really the places where true containment is being moved into an enclosure that's going to be trapping that particulate for good until that filter is changed. And there are a couple different methods. So there's inertial impaction, where a particulate flows in and then ultimately hits a fiber on that filter. Interception, where it grazes in the same manner. There's also diffusion, where a particulate might be traveling all over and finally finds itself in contact with the fiber. And then to a lesser extent, there's also electrostatic attraction, where you might have a charged or negatively charged molecule, and it finds a small bit of attraction toward the fiber in that HEPA or ULPA filter. So how does all of this happen? So particulate capture, as particulates enter and they go to that bed in a HEPA or ULPA filter, which is made of borosilicate glass, it's going to go through a couple different ways. There's a lot of different information on this, but to the short answer in how particulates are captured is that as they enter, they're largely reliant on hitting that glass bed and capturing onto there rather than passing through like a vapor. So it's a physical contact with that bed of glass fiber. So there are a couple of different methods for HEPA filtration or ULPA filtration. There are many different grades and ratings for these HEPA filters, but in short, you want to make sure that you have a HEPA filter that's rated for high capture. And so if we look at the components of each one of these types of filters, on the left you got HEPA. It's made of borosilicate glass fibers. On the right, ULPA is as well. The difference is that the bedding of those fibers is more dense. So if you look at efficiency on the HEPA side, we know that at a particulate size of 0.3 microns, we'll capture 99.997%. And the ULPA, because there's more dense material, it's going to be 99.9997% at an even smaller particulate size. Now, what's MPPS? MPPS is the effective measure of the worst particulate that you could possibly challenge a filter with. So the most penetrating particulate size in this case is 0.3 microns, which means that anything that's going to be smaller than 0.3 microns is going to have a higher containment rating on that filter, and anything that's higher than or larger than 0.3 microns on the HEPA filters will also be trapped much better. So at the very worst, this filter is going to be operating at a very high level of 99.997%. The same concept is true on an ULPA filter. So if you're handling particulates that are incredibly small, say 0.3 microns or so, or even smaller as you go down to nanoparticulates, keep that in mind when you're choosing a hood that has this particulate filter in it. Oftentimes, a HEPA filter will do a good job, but a more densely packed ULPA filter will be more suitable for containment. Once you get to the containment part, there's also the important part of removing that filter. So there's an easy way to do this that prevents yourself from exposure during that change out. Now, this is something that a certifier will do. So if you go into a nearby lab in your facility and you look at a sticker on the hood, there's probably something that says service performed by X company, service was done on this date and met this requirement. Those are the same types of people who should be tasked with changing these filters out. And when they go to do that in LabConco enclosures, they'll find mechanisms to remove those filters into a bag-in, bag-out setup. What this allows you to do, or allows the certifier to do, is to pull that filter out into a bag, keep the inside of the enclosure contained, capture all the particulates and the hood on the filter into that bag, and protect yourself from any unintended exposure. This is a really important part because if you think about your home furnace filter, if you're like me and perhaps in college maybe forgot to change that filter out every couple of weeks or so, you pull it out and you find it's a little kind of dirty. If those filters in your lab are contaminated with all sorts of hazards, you obviously don't want to be exposed to them in the same way that you might potentially be at home in a furnace filter. So that's the idea behind the bag-in, bag-out setup. The goal here is eliminating risk. So even though you may not be changing out the filter, it's going to be the responsibility of someone in your lab, maybe an EHS expert or a certifier from a third-party group. And so that's an important thing to keep in mind. Protect your environment, protect your people who are coming in to do work for you. So the last thing I'll leave you with is just a reminder. Just because you can't see a danger doesn't necessarily mean it's not there. So with a combination of some efforts from Matt's group, certainly finding out, understanding the containment level of hoods that you're employing, and as you continue on, developing new novel nanoparticulates, keep in mind that just because you can't see it doesn't mean that hazard's not there. We try to best iterate that by looking at an iceberg, classic example of Titanic. Small iceberg, big problem. Alrighty, I'll move it over to questions now.
Thank you, David and Matt, for your presentations. A quick reminder for our audience on how to submit questions. Simply type them in the Q&A box found by clicking on the green Q&A button at the lower left of the presentation window. David Wasesha and Matt Hall will answer as many questions as time permits. And the first question is for you, Matt. And it is there a way to do QC on nanoparticles as they change their properties?
That's a good question. So I think it really depends. If you're talking about the manufacturing environment, that there's one answer for that, and then there's another answer for when they're released into the environment. So in the manufacturing environment, there are a lot of different nano characterization techniques out there that one can apply to look at changes. Zeta potential, for example, looking at changes in surface charge, electron microscopy, looking at differences in particle aggregation. So as you progress from maybe having individual monodispersed particles to having more aggregates, there are techniques for doing that, but it will vary. You should understand that that's a very intense area of research, and a lot of effort goes into that, and a lot of the different it's going to be very situationally dependent. Hopefully that answers your question. If there's more specifics around exactly which area of QA/QC you're interested in, please add that in to the questions.
Great. This next one is for you, David. What is the difference between the Expert Nano and the Expert Standard enclosure?
Sure, yeah, another great question. So in our standard Expert enclosures, they've got a similar frame to them, so they're designed with the same airflow characteristics and airfoils, but internally they have a HEPA filter in them. So that HEPA filter may not be sufficient for ultrafine nanoparticulates. So in the Expert Nano, it's got a ultra filter instead with a little more density, so you can trap finer particulates. Another key difference is that the inside of the Nano enclosures, it's an all stainless steel liner. So the work surface, the sidewalls, the back baffle, everything in there is stainless steel, which just helps for the decon process. Whereas on the Expert, you have glass and steel, and those are certainly easy to decontaminate and will last. But making sure that you've got that stainless steel means that you can hit it with some hard cleaners and clean everything up quite quickly.
Okay, this next one, David, would you take it first? What is the best way to decontaminate a device to make sure no nanoparticles are left in the engineering control, and what to do with the HEPA filter afterwards?
Sure. So first thing to keep in mind, anytime you're decontaminating an enclosure is before you reach into it, you want to make sure that the unit's on, so it's going to be under negative pressure. And then moving over to cleaning up for nanoparticulates, that's going to be largely dependent on what you're handling. But most commonly, a broad range cleaner for nanoparticulates will be used, along with some swabbing techniques and tools. A good source for some of that is to look at clean room containment and cleanup devices. Oftentimes they look like very similar to like a Swiffer wipe, but it's best to use one of those tools, wiping down the surface as much as possible and containing that into a hazardous bag. And then obviously removing that hazardous waste bag. Good practice is to double bag it and then pull it out of the enclosure once the entire environment has been cleaned up.
And I'll add some degree of uncertainty into that question. Sorry, David. There's
sure.
The challenge with it actually refers back to what David said in terms of containment. There's what you see, and then there's what you can't see. So verification of decontamination in the nano area is very important. So you could wipe up the surface, you see nothing, but there could be a large number of nanoscale particles remaining on that surface, and that is another area of research where folks are trying to figure out the best practices for what works best for different types of materials, as Dave indicated, and then also how do we verify that a surface has been decontaminated effectively.
David, this next one's for you. Could you please talk about the difference between a Class 2 biological safety cabinet and these cabinets?
Sure. So a Class 2 biosafety cabinet is going to have a sterile downflow air from a HEPA filter, and then it'll go back through another HEPA filter before it exhausts either out through the building or back into the room. So these devices are good containment devices. Class 2 biocabinets are what you think of when you see a microbiology lab or, say, somebody handling a hazardous microbiology. So they are good containers, but they have not been tested suitably against nanoparticulate escape in the same way that our nano enclosures have. So until further testing is done on those hoods, we don't have a good idea of how good the level of containment is for silicon dioxide using nanosafe methods. So I will say this, though. They are internally, the downflow air is sucked into a negative pressure channel back into a filter. So they do have the same control over particulates. It's just in a different method where in a biocabinet the air is coming straight down And in Expert Nano, the device will be pulling the particulate straight to the back wall, into the back baffle, and then into a filter. Another important consideration to make, too, is HEPA filters are common in biosafety cabinets. They're not standard as ULPAs, so if you're working with fine particulates, you would need to switch out the filters in a biosafety cabinet to an ULPA filter. And then it is also uncommon. You can buy them, but it is uncommon to get a bag-in, bag-out system on a biosafety cabinet that's a Class 2 system. So that's something that's standard on a nanoparticulate enclosure such as our Expert Nano.
David, what type of certification is required, at least annually?
Yeah, so the way hoods, whether it's a fume hood or biosafety cabinet or a small balance enclosure, the way they're certified depends on the style of cabinet. In terms of a biosafety cabinet that's typically, or a nano enclosure like this, it's a measurement of the airflow going into the cabinet, a measure of the aerosols going, a challenge aerosol going through the filter. So a certifier will literally come in and generate a test particulate to challenge that HEPA filter, and then they're going to scan on the far side, on the downstream side of that filter to test for leaks. So that's a good practice to do every year. Sometimes, depending on your environment, you may need to do it more often, but it's in short measuring that the hood is going to be containing well through proper airflow going into the cabinet, and proper containment on that HEPA or ULPA filter.
David, what standards should these enclosures be certified against for annual testings?
Yeah, so there's currently a document being developed by another group called CETA for containment testing on small CDEs or Class 1 type powder or particulate containment hoods. That will be a good resource. It's CAG 011. But besides that, the best thing to do is look at a modified ASHRAE test for inflow, and then, of course, for NanoSafe's guidance on testing for nanoparticulate escape through these hoods. And Matt can probably, I'm sure he can provide more information on how to go about finding more information on those test procedures.
Matt, this one's for you. Are you suggesting that the properties of nanoparticles are closer to gases?
I think what we're suggesting is they're right in between gases and the smallest particles that you can see. You see behavior on both levels. In some ways, their behavior can be like a gas, and in some ways, it can be like a particle. And I would say that is figuring out exactly where those cutoffs are going to be is a real challenge.
Okay, this next one is for David. You stated to protect the operators, use a negative pressure biological safety cabinet. What is your need to also protect the product, like trying to keep the product sterile? Do you ever see the biological safety cabinet under positive pressure?
Yeah, so if you're looking at a Class 2 biosafety cabinet, which I did not cover in my slides, you would have sterile downflow air coming out of a HEPA filter or an ULPA filter should you swap it out. That would give you downflow protection for the product. You could use that also for containment because a Class 2 biosafety cabinet will provide containment. But again, we haven't tested those with Matt's NanoSafe group. So if you're to work with sterile devices or a sterile nanoparticulate, that's a little bit of a challenge right now. If you want an enclosure that's also got a NanoSafe listing to it. There are a couple other options. There are things like clean benches, but again, clean benches don't have any way to contain them, the particulates that would be handled in them, so those aren't recommended for any kind of hazard, such as a nanoparticulate.
Okay, so this next one is for both of you. Matt, maybe you will tackle it first. If one is producing nanoparticle waste mixed with lab animal BSL-2 waste, what would be the hierarchy for waste disposal, biological or chemical?
I think that's a great question, and I can tell you, well, first off, I would say in the labs I've worked in here at Virginia Tech, we would try to segregate those wastes as much as possible so that you can manage each waste as specifically as possible. But I would hesitate to answer that with any authoritative guidance. I would say I would defer to the more hazardous agent in this case, probably the biological agent. There's probably a greater concern around that, but I definitely would not want anyone to misinterpret what I'm saying there as any type of formal guidance on that issue. You should probably work through that specific case with one of your occupational safety and health officers to sort through that.
David, could you tackle it too?
Yeah, I'd just reiterate the same approach as Matt. Certainly, it's going to depend on what the particulates are and what the hazard levels are, and ultimately we would make the same recommendation to defer to on-site EH&S.
Okay, Matt,
I will say that it is a complicated problem.
Yes.
Matt, this next one's for you, and the person is asking, not sure if it was slide 24 or slide 25, showed stratification of particle concentrations by height inside the hood. Can you revisit? I don't understand this rationale.
Sure. I'll pull up the slides really quickly. Can I take control here? And I'll go down. Oops. Okay. Looks like 25 or 24.
Okay, 24 yes. Or 25, they weren't sure. Stratification, particle concentration.
24. So I pulled the slide up for everyone to visualize. The rationale here is actually it began as a curiosity to just look at the vertical distribution. So this is inside the enclosure, looking at where particulate levels are most concentrated. So the unit that we tested had a fixed sash opening. So if you picture yourself interacting, if you look at that graph and the face of that, or the area of that graph is what you're looking at sort of in terms of particle concentrations within that enclosure, the sash opening is going to be near the bottom. So that it's a good thing, I think, that you have lower particulate concentrations at that level. However, some of these units have sashes that are movable, so you can raise the sash up to a higher level. And I think that this graph can give you an indication of where the higher particulate concentrations are in that enclosure and what the implications may be in terms of work practice. If you're raising the hood up higher, you may be opening up the enclosure to having higher concentrations of particulates, maybe more accessible to the face of the unit. So it was more an experimental curiosity that we think has some value in terms of work practice.
Now, would you want to click on that slide one more time? I'm not sure if the audience could see that slide, just for a quick second, and then we'll move to the next question. Okay, great.
Okay, is it up there now?
Yeah, good. Alrighty, and this next one is for David. How do you verify cleaning or decontamination?
Well, again, as Matt kind of reiterated earlier with cleaning, so I discussed some just typical expectations for how you would decontaminate, wipe up what you can see. Verification, though, for a clean hood is a little bit difficult, though, because you can't see it. So this one maybe Matt can also help, as well as how you might verify a non-visible contaminant that may not necessarily be able to be chemically analyzed. If you have a powder or something like that, you could certainly swab and then dilute and inject on something like HPLC or LCMS setup and measure. Nanoparticulates, I'm not sure, though.
Just to add on there, we've done some additional testing in a different product line at Lab Conco where we did a fail-to-safe situation where we could actually watch the particulate levels go down as the enclosure was evacuated. So you get this nice slope. You can actually see where the particles go down to levels where you can no longer detect them beyond, above background.
Right. Yeah, and that would be okay for the environment. For the actual surface in the hood, though, for things that are not aerosolized or airborne, a little more challenging, certainly, without being able to have, create a sample for a detector to read.
Yes. I'm sorry. I was talking about the aerosol within the environment, within the enclosure. But on the surfaces, you can look at. There are analytical techniques that you can do where you swap the surface and then you can look at what's remaining, and it will vary. The exact analytical approach would vary based on the elemental composition of the agent that you suspect is present, and it could entail a suite of techniques if it's multiple contaminants that that one would think is present there.
David, how different is nanoparticle and nanomaterial safety and containment?
Yeah, so I use them interchangeably in this case. Any discussion about nanoparticulates and nanosafety, I simply mean as the characteristic of a group of nanomaterials. Just semantics in my behavior, I guess I should say.
David, this next one is for you. What is the airflow rate inside Expert Nano enclosure, inside the Expert Nano enclosure?
Yeah, so we recommend a range of up 60 feet per minute of linear inflow all the way up to 100 feet per minute of linear inflow. In that range, that's where you're going to find best containment. And anything that is going to be of a nanomaterial characteristic, though, provides a real challenge when you have a high airflow. Much like a lyophilized pharmaceutical powder, it's going to potentially disappear off of a spatula if you're running the hood at a higher inflow rate. So typically lower is better as long as you're still within a containment range.
And we have another question from this same person. Can we handle very high surface area materials inside it, like metal organic framework or graphene?
Do you want me to address that one, David?
Yeah, please.
I would answer that as we tested silicon dioxide spheres of 23 nanometers in diameter. I noted earlier in the presentation that we simply could not, it would be prohibitively expensive to be able to test the whole range of materials. But you do raise a very interesting point about issues like high aspect ratio and that sort of thing. And I would say that the testing methods are not there to give you definitive guidance on that right now. And just like this SiO2 aerosolization method is what we think a better step in the direction of getting more accurate information about the containment efficacy of these devices, there are probably additional tests with high aspect ratio or higher surface area materials that one might be able to use on these types of devices to verify that specific scenario.
Great. David, this next one's for you. Who certifies the nano exposure hood? For biological safety cabinets, the NSF standards are applicable. What standards are applicable to nano enclosure hoods?
Yeah, so great question. Class 2 biosafety cabinets have a well-defined test method for containment and operation through NSF. NSF Chapter 49 is the chapter that outlines that. Because of the relative newness of containment devices for nanomaterials, there exists a little bit of an industry problem with a way to test these properly. So you can certainly test them in the best methodology is a modified ASHRAE for inflow, and then also for measurement of particulates on using airflow metering devices if they're not nanomaterials. But when it comes to nanomaterials, it's very difficult. So that's kind of really where Matt's approach to sensing for containment practices is really based out of. And some of the work he's been leading through IEST to develop a standard for testing these enclosures is really what's out there right now and in progress to address that lack of a true test method for nano hoods.
This next one's for both of you. Maybe, Matt, you want to speak to it first. The person is looking for follow-up clarification. Could you discuss nanoparticle circulation studies in animals?
I think you're really challenging me here. Does this one have to do with the waste mixed waste stream question? Was this the same person?
You know what? There have been so many questions. I can't tell.
Okay.
If it's the same. I apologize.
Okay, I'll answer it. I would assume that is the one it's talking about. Maybe they're injecting these animals with these nanoparticles, and then they're trying to figure out how to dispose of that. I would say it's probably beyond the scope of the webinar, but I would be happy to talk with that individual. If they can get my contact information, I'd be happy to talk with them about how they might handle that specific situation. Because I think it would be quite different from some of the discussion on the nanoparticle containment within the enclosures.
David, the next person asks, is my fume hood an acceptable containment enclosure for nanoparticulates? So
in general, I'll take this one, Matt. In general, fume hoods better than working on the open bench. They are operated in a negative pressure device. So for the operator handling materials, they do provide a containment benefit more than the open bench. Problem with fume hoods, though, is a couple of major things. The first one is, okay, well, if I've drawn air away from me and it's pulled the nanoparticulates away from me, where are they going now? In a fume hood, there's typically a straight duct run that goes to some kind of remote blower. And so any nanomaterials that will go past the hood into the ductwork are now contaminating the ductwork, the blower, and then potentially anything surrounding the area where the blower ejects onto the roof. So maintenance workers on the roof, or potentially even on the parking lot where you park every day to go to work at the lab. So that's the biggest problem there, is that they lack a way of capturing those particulates before ejection into the environment. And that would be done on a HEPA or ULTA filter. And so that's the big one. Other auxiliary things to consider are sometimes fume hoods have very high phase velocities that can cause some turbulence. They have a sash opening that sometimes can be wide open depending on user preference, so that can cause containment issues as allows for potential swirl back into the respiration area of a user. And then oftentimes they don't have some added benefits like they don't have ability to have an ionizer built in. Infrequently, are they made out of stainless steel? Usually they're fiberglass or coated steel or potentially PVC, which are just a little bit more challenging to clean up as they carry a little bit of a static charge more so than, say, metal or glass.
Well, I would like to once again thank David Wasesha and Matt Hull for their presentations. Do you have any final comments?
I'd just like to say thank you, everyone, for your time. If you have more questions, certainly submit them, and we will follow up with you and make sure that you get your questions answered.
Same here. Thanks for the opportunity. It looks like there are some really great questions, and yeah, it's a complicated area, and we're happy to help answer any other questions you might have offline.
Great. Well, thank you both again. I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today will be addressed via email after the webinar. We would also like to thank our sponsor, LabConco, for underwriting today's educational webcast. This webinar has been approved for continuing educational credits. Please click on the CE button at the bottom left corner and follow the process to receive your credits. This webcast can be viewed on demand through March 2018. LabRoots will alert you via email when it's available for replay, and we encourage you to share that email with your colleagues who have missed today's live event. Until next time, goodbye.